Hey guys, welcome to the second episode of the mRNA trilogy. This episode will be talking about the history of mRNA vaccines, and it'll include things such as when mRNA was discovered, who discovered it, and important developments in the vaccine. If you haven't already watched the first episode, it was about where I explained what mRNA is and how the vaccines are developed. Be sure to check that out because I think it'll help quite a bit with context for this episode. Alright. So the history of this technology is actually quite long and it's quite complicated. So I'm going to be cutting out a few parts, so forgive me if I don't mention any certain people or certain developments. Now, contrary to popular belief, this technology was discovered in the early 1950s, and that's quite far back. That's about, 70 years ago? So, two researchers had to prove the existence of mRNA, actually. Because remember, during this period of time, the technology that we have today, it wasn't as developed back then. So people had to actually prove the existence of mRNA. Two researchers called Francis Gross and Francis Jacob and their team had to conduct lots of experiments to actually prove the existence of mRNA. Finally, research on how to deliver mRNA into the cells was discovered in the 1970s. Now remember, in my first episode I talked about how injecting pure raw mRNA into the body doesn't work because the body instantly breaks down the mRNA, and there's just no point then. So advances in nanotechnology helped scientists discover that if they use these things called lipid nanoparticles, or nanoparticles made of fats, they could encase the mRNA, the programmed mRNA, in these lipids so that when it reached the cell, it would just break down the lipid casing first, and the mRNA would be able to go into the cytoplasm of the cell and therefore produce the proteins. Then, in 1984, a team of researchers consisted of Paul Krieg, Douglas Melton, Tom Maniatis, and Michael Green and their team used RNA synthesis enzymes from a virus to produce actual live mRNA. And this was to further test the mRNA because, it was a relatively recent discovery, and because it's not that easy to actually get the mRNA straight from the body, so scientists had to find a way to actually create the mRNA in a lab. Then, in 1987, a person called Robert Malone mixed strands of mRNA with droplets of fat, which the human cells absorbed and made the proteins. And these experiments worked. However, as science always is, it's never that simple. And so the mRNA would sometimes still be broken down despite the lipid nanoparticles encasing it. And so then scientists were like, okay, what if we added something to the mRNA to create an almost hybrid mRNA in a way that it wouldn't be broken down by the body? And that's exactly what this Hungarian-American scientist did, Katalin Karikó. She was someone who was really passionate about mRNA technology, and she felt that it really had the potential to develop into something life-saving, and she was right. And so in the 1990s, she worked tirelessly to try and receive funding to research on how to get mRNA vaccines to work in humans, because in 1990, the University of Wisconsin had published a paper saying that it worked in mice. So why wasn't it working in humans? And so she worked tirelessly for years until finally, in 2005, she and her colleague Drew Weissman discovered how to create a hybrid mRNA that was able to reach the body cells without being broken down. And this was by adding modified nucleosides. And now in my first episode, I talked about how mRNA is a single-stranded molecule, whereas DNA is a double-stranded molecule. Now these little things here, that's a nucleoside. And the nucleoside they're chemicals. And each of these, it's like an acronym. Each of these stand for different chemicals. And they each sort of determine certain properties of the mRNA. And so by adding these modified nucleosides, it caused the body to not break down the mRNA. And that was a lifesaver, and it really helped a lot of the issues that were surrounding the research of this technology. And this research was a breakthrough in the scientific community. It caught the attention of two scientists who would later found BioNTech to make a new source of embryonic stem cells. Now, these stem cells are cells that are typically used to regenerate organs and tissues, but that's a story for another time. Then, in the late 1990s, a proposal was submitted to use this technology against cancer. The trials failed, but more and more researchers, including the founder of CureVac, became more interested in this technology. And later on, someone called Peter Cullis developed lipid nanoparticles that reduce toxic effects of RNA in the blood, which allowed it to be stored for longer and maintain stability in the body. And then when the COVID-19 pandemic hit, Moderna instantly started making prototype vaccines with this technology. And the development hasn't just stopped from there. Vaccines have now been found for Zika, flu, rabies, and CMV. However, now we have to ask ourselves the question, why wasn't this technology used to treat so many other diseases? And that has to do with multiple reasons. Number one being the RNA itself. Now the RNA, it's unstable, it's inflammatory, it's hard to work with, it's prone to degradation, and it's expensive to make. It's really sensitive, and it has to be kept in freezers of almost minus 80 degrees Celsius, and that's really low. So especially in countries that have hotter temperatures, it's much harder to transport. Also, this technology wasn't really considered to make vaccines, but it was initially considered to study proteins. And also another thing is the cost of the production. Because if you're a firm, you would want to invest in a technology that that's low risk, that's not very high risk, and would be able to be quite widespread and actually used by the public. And this technology, as I said before, it was mainly used to study proteins and wasn't really considered for a vaccine. That's why firms were quite reluctant to actually invest in this. And that's a brief history about mRNA vaccines and mRNA technology. It's crazy to think about how just a few years ago, almost none of us had heard the term mRNA. And yet now, it's something that we're kind of familiar with. I mean, we injected it into our arms, and as I said multiple times earlier, the pandemic has been devastating, but it's paved the way for so much new development in technology, and without this pandemic, honestly, mRNA technology was not talked about, barely. And yet, after billions of dollars were spent into research and time and money and effort of hundreds of researchers, now we finally have a technology that could be developed into something that could really save people's lives. And honestly, I think that's really amazing. Thank you so much for watching, guys, and I'll see you in the next and last episode of the mRNA trilogy.